Hello, everyone. Welcome to today's webinar. We are really glad you're here and we are ready to get started. Today, we are going to share with you information about the Critical Path Institute's Translational Therapeutics Accelerator, or TRxA for short, as well as details about eligibility criteria, the process for applying for a TRxA award, and much more. We have reserved plenty of time for questions after the presentation, so we hope you'll be able to stay with us. Before we begin, a couple of quick housekeeping items. We have muted participant lines to reduce background noise, but if you have any questions for the TRxA team, and we hope you do, please post them in the Q&A using the Q&A feature at the bottom of your screen. We are recording today's event and we'll make it available online in the coming days so you can share the link with your colleagues. So without further delay, let's get started by introducing you to the TRxA team, beginning with our Executive Director, Micah Everts.
Thank you, Michelle. It's wonderful to be here. Good morning, good afternoon, good night, wherever you are in the world. We have quite the international audience here. Quick background about myself. I'm a Dutch native, so I had my PhD in pharmacokinetics and drug delivery at the University of Groningen. Then I went to the University of Alabama at Birmingham for a postdoc in a gene therapy center, ended up staying on faculty, ended up going through the ranks as a full professor. The last 10 years, I led an academic drug discovery program called the Alabama Drug Discovery Alliance. Had a bit of a detour, worked for 3 years for a contract development and manufacturing organization called Evonik. And last April, I joined the Critical Path Institute to start the TRxA program, and it's been a really wonderful opportunity and experience. So we just had a new team member join, actually, Mark Drew. So Mark, maybe you can introduce yourself.
Yeah, thank you, Micah. So I'm a recent addition to the TRxA team at C-Path. I'm a medicinal chemist. I'm also a patent agent as well. I spent the bulk of my career in pharma at Sanofi Aventis and a couple of stints at biotech at Array Biopharma in Colorado and Icogen, which is at the site in Tucson, Arizona. But I'm really looking forward to discussing the opportunities coming out of academic labs and progressing drugs to the clinic. Okay, thank you. And Michelle?
Great. Again, welcome everyone. I am Michelle Morgan. I'm the Associate Director for TRxA. I started with C-Path in 2018 with our Predictive Safety Testing Consortium. And prior to that, I worked for 30 years at the US-based national office of the Muscular Dystrophy Association. And that's where I really fell in love with the power of research and how it can impact the patient community and families living with all types of diseases. So it's a pleasure to be here with you and to share information about TRxA.
And then last but not least, Kyla, also a new addition to the team.
Hi, everyone. My name is Kyla Edding. I recently joined the TRxA team at the beginning of January as the new project coordinator. I'm excited to be here and to share what we have with you today. I'm going to pass it back to Micah.
Great, thank you. So just wanted to have everybody get a sense of who's who in the TRxA team, and now we'll dive into the details of what we are all about. So we are an accelerator, Critical Path Translational Therapeutics Accelerator, or TRxA for short. So we are a global drug product accelerator, so we have no geographical boundaries. We can fund projects in Alabama, in Tucson, in Paris, in Amsterdam, or in Singapore. It doesn't matter. But what we want to be uniquely known for is the quality data packages that we help put together, as well as regulatory strategies that we help identify of the projects that we support. Ultimately, the goal is to facilitate favorable licensing opportunities between academia and industry. That speaks to one of the first points I wanted to make. We are supporting academic projects, so unfortunately, no startups. And we'll speak a little bit more about why that is the choice that we made. I wanted to speak briefly about the valley of death. So as many of you may know, there's a gap, a valley of death, as it's called, between where academic research typically stops and where industry would like to pick it up to take it forward to the clinic. There are a lot of reasons for that. There's some that are illustrated at the bottom of this slide, and this is from a really great paper. The reference is on the right bottom corner. Wanted to highlight a couple of things that are called out in this paper. So for example, the expertise. What expertise do you need to kind of cross this valley of death that's maybe a little bit different than is typically found in a university laboratory? So it's really a multidisciplinary sport. You need somebody that understands biology, but also chemistry, regulatory science, pharmacology, manufacturing, regulatory affairs. So that expertise is sometimes not so easy to find in an academic environment. Also, intellectual property is quite a big concern. There's a bit of a tension between academics wanting to publish their work so that the society at large can benefit from it. Whereas for an industry slant, you want to make sure that you protect your intellectual property. So at some point when somebody needs to invest probably billions of dollars to get something to the clinic, that they can make their money back. So there's ways to overcome this tension, but it's not something that is pretty standardly done. So you need to think about this very carefully and approach it very systematically. Funding is an issue as well. And of course, making sure that you're on the right path to get as quickly as possible to the patient and try to avoid going on detours. So this is where TRxA then comes in. So at the left side of the slide, let's start there. I just talked to you about the drug development valley of death. It can also be that there's a lack of understanding of the data package that is needed for something to be of interest to industry partners. So an academic professor might think, I just published this in Nature Medicine. I have this wonderful compound that treats asthma, or pick your favorite disease. What else might you need? Whereas an industry partner may look at it and says, well, did you look at how the compound gets metabolized? Does it get into the brain? What are potential safety issues? So there's a bit of a gap there. The other aspect is the regulatory aspects of drug development. So many of us who've been in academia for a while, especially in the pharmaceutical science, the big picture. You go from preclinical research to clinical trials, phase 1, phase 2, phase 3, then ultimately you get approval by the FDA. But the details, that's really where sometimes the knowledge is lacking. What is your ultimate patient population? What is the readout? How are you going to figure out if your new therapy actually works well, yes or no? All those aspects are, again, something that's not typically on the radar of a university professor. And then the last point here is that sometimes academic studies lack reproducibility. There's been a lot of publications around this in the last few years. Numbers vary from 30% to 80%, depending on what lens you look at it. It's a concern just so that an industry partner will want to repeat the work that you may have done in your lab in their own hands to make sure that it's actually working. Well, if you already have somebody else, a third party, reproduce your study, that adds to the value of the data package that you might bring to bear. So then in the middle, the solution that we want to offer is for TRxA to provide both funding and expertise. That expertise comes mainly in the shape of regulatory guidance, as well as the assistance of study design and implementation. What experiments do you need to do to get to the clinic as fast as possible? And then the third point is that we will coordinate involvement with CROs or contract research organizations to maybe validate the academic findings. So your key experiment, let's have that done in a different group, for profit. They have no skin in the game what the result is, except to do their best research possible. If it then shows up as the same result as you have, that really adds a lot of value to the package that you then go to the industry with. And then there's parts of work that are just not typically done in academic research labs. Now, it differs a bit from lab to lab, but for example, if you have a biology lab, you may not have chemistry resources where somebody can make compounds for you. Or you want to do pharmacokinetic studies. Well, it's sometimes done more quickly and more cheaply in specialized companies who can have a really quick turnaround for that, instead of you setting that all up within university boundaries. Or maybe there's things that are just not typically done at all in university labs, which is, let's say, a good laboratory practice or GLP study in dogs. So the more we can outsource these kind of parts of the process, hopefully the more smooth we can make it, and we will really help you with that engagement, again, to get something to the clinic and to patients as quickly as possible. So I wanted to point out a couple of things on the right-hand side of this slide. We are an independent nonprofit. So we are a neutral facilitator, and we'll talk a little bit more about the Critical Path Institute as a whole that really influences how we also as TRxA kind of view our role in drug discovery and development. We will not claim any ownership of intellectual property that you develop or projects that we support. So that will probably be of interest to your technology transfer office. So we will leverage the Critical Path Institute's regulatory knowledge and relationships, and I'll tell you a little bit more about that in a couple of slides, but then also provide strong project management to facilitate streamlined planning and execution. So this is not a grant where you get a check and then a year later, you give us a progress report. It will be really a mutual, project team that we set up, really a collaborative approach to get the project, again, to IND, to the clinic as quick as possible. So let's look at the bottom of this slide here for a second. So that is a pipeline of the typical description of drug discovery and development. And I wanted to highlight where TRxA wants to play. So we have indicated lead optimization through IND enabling studies. So that means that we do not fund any earlier stage discovery studies. We will not fund high throughput screening or hit validation type work. We also do not fund anything in the clinic, but at in-between space, and that's really where this valley of death has a big gap. That's really where we want to have our sweet spot and support projects. So we are funded ourselves by another nonprofit called the Frederick Gardner Cottrell Foundation. And this was established by Research Corporation Technologies, which is a venture firm in Tucson, Arizona. They established this foundation many moons ago, December 1998, and they provide financial support for scientific research and educational programs at nonprofits like the Critical Path Institute, or C-Path. So let's talk a little bit more about the Critical Path Institute as a whole, or C-Path. So again, this is our mother organization, not TRxA itself. So C-Path was established in 2005 or so as a result of the Critical Path Initiative that was instituted by the Food and Drug Administration. So we act as a trusted, neutral third party to facilitate public-private partnerships. And you can look at the graph on the right at who all plays in these partnerships. So there are a lot of industry members, and they actually pay a membership fee to be part of these partnerships. Then the regulatory agencies like the Food and Drug Administration, but also Europe's EMA, as well as Japan's PMDA. Then we have the voice of the patient that's really part of this, which is proven to be such a critical part. And if you discover and develop drugs, actually talk to your ultimate patient to see what they need and what their request is really quite critical. Then we have other stakeholders, like, for example, the National Institutes of Health in the US, and also academic research institutions all around the world. So what happens in these partnerships is that we enable iterative, so continuous participation with the regulatory agencies to develop new methods to assess safety and efficacy of medical products. I'll show you in the next slide some examples of those, but just to give you a bit of a teaser, for example, biomarkers or clinical outcome assessments. And then the goal is to have official regulatory endorsements of these new methodologies and drug development tools, or DDTs. That way, if such a tool is developed, then anybody can use it, go to, let's say, the FDA or EMA, says, hey, I want to do a clinical trial in this particular therapeutic area, and I want to use this tool or this instrument to measure if my drug has the right effect or not. Because the regulatory agencies have actually seen this, they help co-develop it, there is a confidence, so that gives confidence for a lot of money that's being invested that this is going to yield the right result. So we convene these scientific consortia with industry partners, academia, government, all together to share data and expertise. So really get together the best science, broad experience, build consensus, and then share the risks and the costs around that. So this is a diagram then of C-Path's concentration areas and core competencies. So at the top, you can see the little badges of our core competencies, and those include data management and standards. So we have a data collaboration center, for example, where pharmaceutical industry deposits clinical trial information in those databases so that everybody can access them. It's all de-identified, it's all made anonymous, but still, and there's a lot of science in how you treat data so that everything can talk to each other, things like that. We have quite an effort in biomarkers as well. And you can think of that as maybe early indications that your drug is working without waiting for the ultimate final end goal of, that are longer term outcomes. You can also use it for safety prediction. If there's a signal in an animal model, what kind of biomarkers can we look at that predict whether something's going to be safe in humans, yes or no? We have a big modeling and analytics group, a quantitative medicine group, They do, for example, the development of clinical trial simulation tools. And investigators can use these tools to dive in, and they kind of build on those data management and standards groups to all the data that's in there and see, okay, if we want to do a clinical trial, if we expect this kind of effect size, how long should the trial be? How many patients should we enroll? And that ensures that you have the right design, so you do it right from the first time, and you don't have to repeat it later because you missed something with your design application. We have clinical outcome assessments. Some examples there are diaries that can be filled out by patients as they're in a clinical trial. We just had one approved, for example, for asthma, where there is a parent diary as well as a child diary, a morning diary, and evening diary. And again, there's so much science in developing those diaries so that you get meaningful information out of there that can inform whether a clinical trial is having a good result or not. And underlaying all of this is a lot of regulatory knowledge, a lot of development science that C-Path has really brought expertise in. So then at the bottom, you see a list of concentration areas. So these are the therapeutic areas that we focus on. We don't exclusively work on those, but they're really the main bread and butter. So neuroscience includes, for example, Parkinson's disease or Alzheimer's disease, as well as a couple of others. Immunology and hematology include type 1 diabetes or sickle cell disease. In an infectious diseases group, we have a large initiative in tuberculosis. Safety science, again, has to do with this prediction of if you see something maybe in a cell culture system or an animal model, what does that mean for the human condition? Rare and orphan diseases and pediatrics. Underneath all of that, the bedrock is that we are a unique neutral convener. So we don't play favorites with anybody in particular. We're really kind of the neutral Switzerland of all those different parties that come together, and we're trusted for this. So for TRxA, tying it back from the large C-Path view to what does TRxA do? So our initial focus area of TRxA will be to support projects in these concentration areas using small molecules. Now, these small molecules do need to have the opportunity to get composition of matter intellectual property. So that means repurposing drugs is at this point in time out of scope. But it's something that gives you some guidance, some boundary conditions to know what we're looking for. Now, you may see like, well, there's no oncology on this slide. Well, again, we give preference to therapeutic areas that are on this slide, but we're not exclusive to them. So if you have the wonderful oncology project, well, first of all, it may fit into the immunology and hematology bucket because there may be an immunology component to your approach. It may even be a rare and orphan disease. It may be a pediatric tumor. So then you actually do fit into these buckets. But even if you don't, I would still encourage you to apply, because if it's the right project at the right stage, we will definitely take a look at it. So then how does TRxA operate in this larger C-Path context I just told you about? So we can really synergize with all the consortia that C-Path has built over the years. There's more than 20 now, I think it's close to 30. These consortia all focus on drug development tools. And TRxA is a new direction for C-Path in that we directly focus on drug discovery and development support. But still, we can tap into those consortia and have all those subject matter experts that we can reach out to, whether they're in industry, whether they're at the regulatory agency, whether they're a clinician, whether they're a patient. We can kind of bring all those people together around the projects, depending on what the project needs, and get that input. So we will apply C-Path's regulatory expertise as well as the project planning and management skills to really move projects to the clinic as quickly as possible. And in this slide, we have the link. Probably all of you are here because you know of our request for applications is live, and the link is here on the website. And I'll show you a little bit more details about that in a few slides. So we have 3 different project types that we fund. We probably need to come up with a more creative name for them, but they're called stage 1, stage 2, and stage 3. Stage 1 is if you have an early lead compound and you want to develop that into a late lead series. So probably you need to do a lot of medicinal chemistry, a lot of testing. We will fund that up to $250,000 US for approximately 1 year, for up to 1 year. Now, if you have a project and you only need something for 6 months, absolutely come talk to us as well. Also want to point out that these dollar amounts are total costs. So that is the direct cost of the project plus the overhead or the indirect cost. The indirect cost is capped at 10% of the total cost. As a nonprofit, this is pretty typical. We just want to make sure that our well-earned dollars go to this project support. Stage 2 is a late lead series to selecting your clinical candidate. And this is when you have a couple of good ones, but you really need to hone in, okay, which compound am I going to put in a human? This will be funded at $500,000, also up to 1 year. And then the last one, stage 3, that is funded at $1 million US. This is when you've selected your clinical candidate and you need to do IND enabling studies. For example, GLP toxicology studies in animals. So let's dive into the details a little bit more. What does it mean, stage 1, stage 2, stage 3? So for stage 1, the entry criteria, so we would look at your project. If your project is an early lead optimization, you have tractable drug leads from ideally multiple chemical series. So your main one and a backup series, that will be very beneficial. And you really need to have demonstrated that you can have optimizable SAR. SAR stands for structure activity relationship, which means that you've shown that you can manipulate different parts of the compound to, for example, make it more potent, more selective, less toxic, more stable, depending on what you need to do with the compound to make it a suitable compound for using in humans. You also need to have established in vitro pharmacology assays, can be a biochemical assay or a cell-based assay. And then you also need to have established an in vivo pharmacodynamic model. So that doesn't mean you need to have shown that it works in an animal. It just means that you need to have an animal model identified in which we can test it later. This is not always obvious. It depends on the therapeutic area. If there's good animal models out there, well, who has them? What is the access for that? So again, for you, it may be really a no-brainer. Of course, I have a model. For somebody else, it may actually be quite challenging. So we just want to have some information around that. What is the animal model you can test this in? The success criteria, so hopefully after a year and $250,000, the result would be that you have a well-defined compound progression pathway that established success criteria. The compound progression pathway is really a roadmap. What are the different steps, the different tests and assays you need to do in which order? And what is the performance that a compound needs to have in order to graduate to the next one? You would have optimized leads from multiple series. Again, not just one good one. Having a backup from a different scaffold will be really beneficial. You need to have characterized in vitro pharmacology properties, including how it works in cells. Characterized ADME properties, standing for absorption, distribution, metabolism, and excretion, in vitro, and also in a rodent in vivo. So it can be a mouse or a rat. And then also demonstrated in vivo pharmacology in a pharmacodynamic model. So pharmacology will be different sometimes than therapeutic efficacy, right? Maybe you just have the ability in a certain animal model to show target engagement. My compound interacts with the target it's meant to bind to, and it inhibits its function, for example. That might be an appropriate endpoint for this. Again, some therapeutic areas may have very difficult and long therapeutic efficacy models, so that can be pushed to a later stage. Again, you do need to have identified, though, what your in vivo efficacy model will be for stage 2. So let's go to stage 2. The entry criteria are really the exit criteria, success criteria for stage 1, so I will not go through those. But for stage 2, what really the goal is here is to define your target product profile and also a regulatory plan to achieve this target product profile. And your TPP, as it's called, is really a description of what you want your drug to do in patients. Is it a once-a-day dosing? Is it an oral tablet, or is it an IV infusion? What's your patient population? Pediatric, adult? There are certain criteria. You need to have thought about these kind of things. So at this point in time, having a late lead is really, we've optimized the molecule, meeting candidate selection criteria. So characterized in vitro and in vivo pharmacology, including now at this point in time, you need to have shown efficacy in an in vivo model. Characterized ADME properties, again, in vitro and in rodent, but at this point in time, maybe also non-rodent in vivo, for example, DocPK. Characterized toxicology properties, also in vitro and rodent and non-rodents in vivo, and then also having defined a non-clinical formulation. Sometimes, especially, well, let's say, depending on the expertise that you have, there's actually more to a formulation than just maybe dissolving your compound in a little bit of buffer. Maybe you need certain excipients that, help with solubility, or maybe to, change the pharmacokinetic properties a little bit, or maybe to help with oral absorption. You need to have thought about these kind of things at this stage in the game. Also, you need to have defined your API, or active pharmaceutical ingredient, scale up and characterization plan. how are you going to make your drug? And that can be very different depending if you need to make 10 milligram on the bench versus, 100 kilo in a huge factory. There may be different steps that you need to do. There may be different synthetic routes that gets you to the right end goal with maybe a better yield. Again, this is something that we need to work on, and we can help with that. Stage 3, again, the entry criteria are the success criteria for stage 2. But here at the end of stage 3, you would have a compound that adheres to the target product profile and the regulatory plan. You really have a well-characterized molecule at this point in time with a complete toxicology package that really enables a first-in-human study. So that means general toxicology, but also safety and genetic tox. At this point in time, you also need to have an optimized GMP API scale-up strategy. Again, making sure that we can make this in big tanks in factories, wherever you are. So let's talk about the process in a little bit more detail. So currently the call for pre-proposals is live. The deadline is April 17th, 2 p.m. US Eastern Time. The link is here. If you go to our website, it will be pretty easy to find. There is a tab there, and I'll show you a screenshot in a second, that says application and guidelines. There's a document there with the PDF that gives you a lot of information, exactly what are we asking for, what are the scientific review criteria, so you can tailor your answers to that. But what we're really looking for here is a high-level description of your project, really a non-confidential summary, like what is your molecular target, what are the properties of your lead molecule? What are the next steps that you think you need to do to get to IND, and then what would you use this funding for? This pre-proposal, again, this high-level summary is reviewed by TRxA's Programmatic Review Board, or PRB. This board determines which projects advance in the process, and this is an internal group of people within the Critical Path Institute. Then the projects that move forward, we will initiate a non-confidential agreement or non-disclosure agreement, confidentiality agreement with you. That is because we want to see the chemical structure at this point in time. So we will negotiate with university to get the CDA in place, and then uploads of the confidential information. So that could be the chemical structure, maybe other confidential information, needs to be completed by the end of June. And then we have a review by medicinal chemist to really look at the chemistry and say, does this have legs, yes or no? If you've been around any chemist, there are usually lots of opinions around if compounds are ugly or great, but we have a collection of medicinal chemists put together that's going to look at these structures and again, make some determination, is this going forward, yes or no? The ones that go forward will get invited for a full proposal. We will send out invitations on July 17th, and then the proposal is due September 15th. So you have about 2 months to put this together. Now, throughout all of this, coaching and mentoring is provided by us, the team that you just saw, so that we can help as much as you like to put together a plan. So some of you will be like, well, I know what I'm doing, I'm just going to submit. Others, you may be a little bit figure out what you need to do. So we're available for consultations, so you can reach out to us and to set up a 30-minute call, and we can give you as much feedback as we can. And that is because we are not the reviewers. We actually, especially at this point in time, we have an external scientific advisory committee who will review and score their proposals. The members of both the PRB and the scientific advisory committee are actually on our website, so you can look up who's doing that. So the scientific advisors really is a collection of chemists, ex-pharma, consultants, some venture capitalists, clinicians, regulatory scientists. So they look at the proposals all with a little bit of a different lens to make sure that we capture the best advantages and disadvantages of each proposal. So once the scientific advisors finalize their scores, those scores get forwarded back to this Programmatic Review Board, or PRB, and they will make the final selection of funding based on programmatic fit. Of course, it needs to be a really good proposal, but as you may know, sometimes you have really a group of really good ones, and how do you then pick? That's where the programmatic fit comes in. And that's also what ties back a little bit to those therapeutic areas that we have a preference for. So only if 2 proposals are equally scientifically strong, then we may give a preference to one that we have an active consortium in. We go to the next slide here. So this is a screenshot of our website. So we're at C-Path.org, c-path.org. If you navigate to Programs and TRxA, you will land on this page, Translational Therapeutics Accelerator. And here at the bottom, you see these tabs about TRxA, funding opportunities, FAQs, application, and guidelines. If you go to that tab, Then there's a link to this guidance documents for applicants. This is a PDF. And then this is actually a fillable form. So I will show you on the next slide what some of those questions are. And of course, you can go to the website and look it up yourself. But we really try to make it as straightforward for you as possible to provide us the information that we're looking for. So here are some of the questions. This is a high-level overview. There may be one or two additional small ones in, but of course, we're going to ask you what's the name of your project and what type of award are you applying for, stage 1, stage 2, or stage 3? What is your therapeutic indication, as well as your target population? So if you're doing type 1 diabetes in adults, something like that, we would be interested in knowing. What's your biological target that you're trying to target with your compound or the pathway? Is there any structural information available, yes or no? And it will be a pop-up field that will say, well, if so, then what is it? Can you give us a link to a database or something? How does the project address an unmet medical need? And I think most of you will be working in this field because, in academia, we always try to solve new problems, but still it's something that you would need to verbalize. What is out there to treat this particular disease? What is, the problem here that you have the solution for? And then also, what's novel about this? What differentiates your project from competitors? So having a sense of what maybe companies are doing, what maybe other academic groups are doing is really helpful to see, okay, so they're trying this approach, but we're different because articulate why this is novel and inspirational. This is really something that our reviewers are going to want to take a look at. If there's any genetic evidence of the relevance of the target, that will be useful. Again, it's not a requirement that there is any, but if there is, we just wanted to make sure that we asked about it. What's the scientific rationale of the target as a whole? Are there any safety issues that we could be concerned about depending on what the target is, or maybe what the structure of your molecule is, if you know a little bit about that? What is the status of your intellectual property? What are the next steps you need to do to get to IND? So it's really the end goal here. But then also the last one is, okay, so then with this funding, what part of that are you going to do? So what are the specific activities? You can see there's the most space there with up to 500 words for activities to do with this funding. But I really would recommend you stay as organized as possible. So bulleted lists, time estimates, that will be really useful in review rather than very wordy, flowery language. So be as crisp and as precise as possible. That's going to probably help you in the review. So then I have a couple of FAQs that I wanted to touch base on. The first one is, well, who's eligible? Who can apply? Well, you need to be a faculty member and you need to be at the university or a nonprofit research institution, but you can be anywhere in the world. And then somebody asked, well, so does TRxA funding come to my lab? And then I pay the CRO. How does this go? Well, actually, we award the funds to the project, not necessarily to your lab. So that may mean that part of the funds, or sometimes even the majority of it, may go to a CRO, this contract research organization, to help you execute the project. But still, this support combined with the information that we provide to put together a target product profile, a product development plan, as well as the regulatory strategies that we help define, really will make your project more attractive for licensing deals and then moving towards the clinic. So what can you use the money for? Well, anything associated with executing the defined project tasks. So if you need to buy specific lab supplies, procure animals, or services, core services that are within your institution or maybe another institution, absolutely allowed. Salary support gets a little bit more tricky. It is allowed, but really only if it's associated with executing a specific task. So if you have a technician who's going to do a 4-week animal experiment in your laboratory, then of course we can fund that technician's salary. But it needs to be enumerated and identified as crisply as possible. What's not allowed is general laboratory expenses. So if you need to buy new lab coats for your team members, find another source for that. We also do not cover travel, equipment, tuition, or IP protection. Especially since we do not have any ownership of the intellectual property that you're developing, it's really the expectation that the institution itself will cover that. And I mentioned earlier, indirect costs are capped at 10%. One more slide. How do I deal with CROs? How do we even know who to pick? Well, we can help with that. We're new, so we don't have black book yet of who the best one is for what kind of experiments, but we do anticipate building that. But we will also, with our expertise, with their network, can certainly help you identify what the suitable CRO is to do part of the work. We will also manage the contracts for you and payments. So we will directly pay them. You don't need to worry about that. And that way it really saves you a lot of administrative hassle. But of course, if let's say you apply for a $250,000 project and $100,000 needs to go to a CRO, then that gets subtracted from the 250K. So you now have 150,000 left for your lab. So you need to kind of think through that when you make your budget. Somebody asked us, well, do I need to worry about overlap with other funding sources? Like I have an NIH grant to study this, right? Can I apply? Actually, we're not worried about that. Of course, you never double dip. You don't use the same money to fund the same research, right? That's unethical, and I don't think anybody would do that. But actually, it may be beneficial if you have different aspects of this project funded by different institutions. So if you have an NIH or DoD or maybe IHI grant to fund, a certain component of your drug discovery platform, and you realize I really need to do this part too, then TRxA can come in and help you with that. So that's absolutely not a problem at all. So what do we get out of it as TRxA? We're not claiming any IP, so we're not going to get rich, right? Well, actually, there is a terms and condition document that we will request in this document a percentage of income to the institution. This is actually pretty typical. Other nonprofits do it as well, so we're not doing anything crazy here. So if there's a success, and only then, and let's say the university gets $1 million up front for a payment, and then future milestones and royalty payments if the project is a success and a pharmaceutical company takes it all the way forward, then we want to have a little slice of that. And that little slice, the size of that will depend on how much we have invested. So there will be a lower percentage if we've invested 250,000 and a higher one if we've invested maybe over $1 million. So we're happy to share this template with you so we can share it with your grants and contracts office or your tech transfer office so that can take a look. Is this palatable to them? We've run it by a couple of academic institutions already, and it's nothing crazy, nothing outside of the norm. And then the last one is, of course, everybody wants to know, well, what are my chances of success if I apply? So how many projects do we fund? Well, it depends a little bit on the stage of the projects because, we can fund 2 stage 1 proposals at 250,000 each, or 1 stage 2 proposal for 500,000, right? So we anticipate funding between 3 to 7 projects per year in the coming years. So we do hope to grow the resources that are available so that we can support more projects. But this is our initial budget between 3 and 7 or so. We have to see. And I cannot give you any statistics yet of how many applications we will get. This is our first global round. So it could be 20, it could be 200, it could be 5,000. My gut feeling is it's probably going to be around 100 or so, but really we can be off by an order of magnitude. So no data yet. The next round, we will certainly be able to give you more details on that. So with that, I'll stop and I will ask Michelle to come back up and help me navigate through some of the questions. And a reminder, put your questions in the Q&A button at the bottom of the screen, and I will try to answer as many as we can.
Great. Thank you, Micah, for that great overview. And you answered so many questions that we typically get, so that was very helpful. And we do have some additional questions that have been coming through. The one question is, do you need to have IP already filed?
I'm glad somebody asked that question. The answer is no. So there needs to be an opportunity for developing IP. So that means that natural products are probably out because you cannot patent anything in nature, right? So it needs to be new composition of matter, but so there needs to be the chance for filing, but if you already filed, that's fine too. If you don't have filed yet, sometimes that may actually be even a little bit beneficial because it means you can work on it a little bit longer and make your patent application that much stronger. So talk to us, at least if there's the opportunity for IP. If you know that nobody else has patented your compound, for example, that would be probably fine. We also want to make sure that intellectual property is covered globally. Now, that doesn't mean in every country in the world, but if you only file in, for example, North America, so only the US and Canada, that's going to be less attractive than if you also file in Europe and maybe some Asian countries as well. So think about that a little bit. Talk to your tech transfer office. Most of them, I think, will file, at least, globally, different countries around the world. But that is definitely something that our reviewers will look at as well.
Great. The questions are really coming in fast and furious now, Micah. So I hope you're ready. Continuing on the conversation of IP, if an academic PI has intellectual property already secured and they've started a startup company, are they eligible for TRxA funding?
Unfortunately not. So we are at this point in time only focusing on IP that's still within university walls. And this ties back into the Critical Path Institute status of neutral convener. We cannot be seen as playing favorites with anybody. So we cannot support an individual company because it may be perceived as giving them an unfair advantage when they finally go to the regulators, because, you know, we know the EMA, we know the FDA, we know the PDMA. So we're shying away from that. Now, who knows what the future brings, but at this point in time, it's academic only, and the IP still needs to be within the institution. So it cannot have been out licensed to a startup. It cannot have been out licensed to a larger pharmaceutical company.
Great, thank you for that clarification. Another question here, are novel diagnostic tools and assays part of the supported portfolio?
Oh, great question. As you might have guessed from the Critical Path Institute overview, we are very much in favor of diagnostics and biomarkers and so on. Unfortunately, that is outside the scope of TRxA. So I feel I'm saying no a lot here. Right now, at least for TRxA, it's small molecule drugs only. So no devices, no diagnostics, also no biologicals or cell and gene therapy. Now, especially with the latter, I can see us expanding to that after maybe 1 or 2 rounds, where we also can support maybe a peptide, an antibody, an mRNA LNP for, you know, a therapeutic cancer vaccine. Maybe next round, not right now.
Okay, great. I'm going to give you a question that you can say yes to. So, will you guide applicants on what stage they should apply for? For example, if research is somewhere between stage 1 and 2, how do they know what to apply for?
Yeah, sure. Yeah, reach out to us, I would say. I would also say, I think we will post this video on the website, plus in the guidance for applications, it also enumerates those entry and success criteria of the different stages. So look at that again, then ask for a consultation for us. We have them in 30-minute increments. We try to accommodate as best as possible with your schedule, and we'll take a look. So it does help us a lot if you give us information up front as much as possible. And I think, Michelle, you're developing kind of an intake form, right? It's nothing fancy, but just give us a bit of a hint of what you want to talk about in some pre-read, so that when we meet, we can actually give you the most meaningful advice and having thought about it instead of, you know, off the cuff.
Excellent. Okay. On the topic of CROs, CDMOs, does TRxA help find these globally? And then how do we work with them in terms of their management?
Oh, thank you. Thanks for clarifying. This is also, we're new, so we're going to feel our way through this. But yes, we will help you find the right CRO. We're not asking for you to have identified them before you even, you know, put in a pre-proposal. So we can probably give you some recommendations to get at least a quote, so that you know, like, okay, I want to do, you know, 10 pharmacokinetic studies with these compounds and, you know, this many rats. You can get some quotes and you can kind of know at least how much it's approximately going to cost, so you can build your budget. So we can help you connect with them. We would then probably set up a 3-way confidentiality agreement of some sort, so that we have a party with the CRO, a party with us, and a party with the university. And then we will pay directly, but you will own the data. So CROs typically assign all the intellectual property to whoever they have a contract with. So we will make sure that it is assigned to you, not to C-Path. And we will work with our legal team to kind of work out all the legal documents. And again, CROs can be anywhere in the world as well. So it really depends on budget, the quality, exactly what you need. There may be some specialized niche applications where there's one group in Stockholm and they are the best. Well, then we'll just deal with them, right? So it just depends on what your project needs and who the best provider is.
Great. Okay. Speaking, we have a couple questions that relate directly to the application process itself that you delineated. And one of the questions is, can academic tech transfer offices apply on behalf of investigators?
Oh, interesting. I would say yes. So as long as you fill out the information, and we probably want to know a little bit about the investigator, but you probably know that, right? But we're not asking for huge CVs. It's just, why is this investigator really suited to be part of this project? So yes, that would definitely be possible. We also, what we can offer is if you have a portfolio, if you have some one-pagers of your projects, feel free to, maybe set up a call with us, or maybe this might even be better via email. Send us like, hey, we have these 10 projects. Can you kind of give us a sense of which ones are kind of thumbs up, thumbs down? We can certainly do that. It will save you time, it will save the applicant's time if it's like, use your time elsewhere. Or we're like, hey, this one will be awesome. Make sure that gets submitted. So we can work with you. Yeah, it's there's flexibility there.
And speaking of email, just for folks on the line, if you want to reach out to us via email, that address is TRxA, TRxA at C- thank you, Micah, C-Path.org. So you can reach out to us with those questions. You can also reach out to request being included on our mailing list so you receive news and updates. Another question on the application process, Micah. Looking forward to step 3, right? The full proposal. Do we have any, can we provide any information on what the format of that would be? For instance, is it going to be something like an R01 grant with just 12 pages of science? What can folks expect?
Yeah, a great question. So I anticipated, this is in flux. we are actually trying to optimize some documents that we've worked on before. So what we want to make sure is that we get very targeted information. So it will probably be a version of the pre-proposal with a very specific question and a certain page limit to how we want to answer that to be. So there's not going to be a blanket, oh, you have 9 pages to convince us that you're awesome. It's going to be very, more in-depth probably questions around, okay, you told us about the IP and you probably answered secured or whatnot. Let's actually dive into that. What does that mean with respect to the science of the target? Instead of, I forgot the exact word count we have. Let's say now we have 100, we're going to give you a page, right? Or maybe 2 pages. So it'll be something like that. So it will be an elaboration on the style that we've introduced with the pre-proposal. The exact format, whether this is going to be an online form or just, put together a PDF, is in flux. We are working right now to see if we can get a vendor to have a software solution. These discussions are ongoing. We hope and think it will be in place at the time of the full proposals. But should that not be the case, then we'll figure out a interim solution. But it will be very specific, directed questions rather than give us 5 pages on research strategy.
Great. Okay, we spoke a lot about the requirements of projects, right, that would be eligible for TRxA funding. What about institutional requirements?
Ooh, well, broad strokes, universities, you're good. Nonprofit research institutions, you're good. Beyond that, if there's something like, hey, I'm special, am I eligible? Just shoot us a note. We'll take a look. So nonprofit is quite critical. But other than that, I don't see anything off the top of my head where I can elaborate on that. So yeah, feel free to reach out to us, give us some more details, and then we'll tell you yay or nay.
Great. Okay. We mentioned that infectious disease is one of the concentration areas, and we called out specifically tuberculosis. Would TRxA awards be limited to tuberculosis only?
No, I'm glad somebody asked that. So it's just an example. We happen to have a lot of infrastructure around that, but we're not limited to that. So if you're working on a viral pathogen, absolutely come talk to us. We've all lived through this pandemic, so we all know there is a huge unmet need there as well. So not limited to tuberculosis whatsoever, or any other bacteria. We have got a fungal problem. Let's talk about that too.
Okay, keeping an eye on the time here as well. One of our attendees indicated they have a druggable target, but not the actual drug. Would that study qualify?
Unfortunately, that is a little bit before where we would come in. So we need to have a drug. There's a couple of other resources that do that pre-part. So one that comes to mind immediately, especially in Europe, is Ligature's European Lead Factory. That may be a resource for you to reach out to. They have a consortium set up, or I don't know if that's the right word, but they pull together different chemical libraries from different pharmaceutical companies, and they will pay to develop an assay that can be used for high throughput screening of your target to find new chemical matter. They will do a little bit of initial optimization, I think, but then they might feed into TRxA. So for us, you really need to have a lead molecule. The earlier discovery phase is not eligible, but maybe look at your tech transfer office for gap funds or kind of innovation development type funds, anything else that it kind of helps at the initial step, and then we'll happily take it once you have some chemical matter.
Great. And we did speak about composition of matter IP, and we have a question here about a method of use patents and how TRxA would see those.
Yeah, so this is why we kind of identified that the composition of matter IP is quite critical. Method of use, when you're really looking at, for example, repurposing, molecules being used for diabetes now can also be used for asthma. That is a little bit more complex business model. We actually within C-Path have a lot of interest in repurposing. So it's not that we're saying like, well, forget about that. That's not. It's actually a really interesting approach, especially for rare and orphan diseases. There's a huge emphasis in Europe at this point in time as well. There's new initiatives that Critical Path is part of. Unfortunately, for TRxA, we're putting it out of scope. And that is because there's a bigger societal conversation that needs to happen. How do we fund that? Who's going to ultimately fund, let's say, phase 3 clinical trial? And that needs to be somebody who thinks that that method of use patent is strong enough to reap their rewards. And sometimes that may be the case. Sometimes it's a little bit more tricky. And because they get so nuanced, we wanted to kind of shy away from that, put it aside for right now. So maybe later, this is something that we can expand to. If you have just a matter of use IP, let's talk. But for this initial round, just to keep things a little bit simple, also for our first go at this, we are really focusing on projects with composition of matter IP.
Thank you, Micah, for that explanation. We have an attendee who indicates that they have a small molecular drug and they know the target molecule, but they do not know the binding site. They have the phenotype in vitro and mouse model, do not have a group of drug with similar structure. How would TRxA look at that opportunity?
Oh, if you know the target and you have a molecule, this is one where I'd say let's talk so that we can look a little bit more detail. What do you have? Because we're really looking at drugs that have optimizable SAR, having at least an inkling that you have this structure-activity relationship that you can manipulate a little bit, will probably rank it higher in the likelihood of it getting picked for funding. So with that one, I'll be less enthusiastic if you only have one molecule and nothing around it. I think our chemists are gonna just scratch on their heads like, yeah, lucky, So not to sound too like a negative Nelly, I don't think that's gonna rise to the top. But if there's special circumstances, of course, we can always take a quick look and give you a final yay or nay.
Great. We spoke a lot about TRxA being focused on small molecules, not funding at this time biologics or drug repurposing. Can we talk a bit about what our criteria are for a small molecule?
Oh, yeah. How small is small? We actually had a conversation about that within the team a few weeks ago when Kyle joined. So small, like really small. So small is typically considered like 500 Daltons or less. Now, if you have something that's 650, probably falls into that realm as well. Peptides can sometimes be a part of that. So within the Alabama Drug Discovery Alliance, we had a peptide project that started as a 4 amino acid peptide. We then, with chemistry, made some unnatural amino acids. We removed chiral centers and it really morphed into a true small molecule. But if you're at that size of a small peptide, then I say also, let's talk. Once you get more than, let's say, 10 peptides, then they're really getting into larger biological territory. And the reason why we're narrowing that a little bit is just because of the expertise that we have on hand. The scientific advisory committee expertise that all tend to have worked in small molecules. Again, like Michelle, like you hinted at, I think we can expand and will expand beyond that after a couple of rounds, or maybe even already after this one. But to kind of, walk before we start running, we said let's focus on small molecules either. Same with what did somebody ask? Like a small DNA or RNA sequences. That's also, it gets to a very different chemistry. So I would say that's not really what we'll be looking for.
Okay, thank you for that clarity.
Mhm.
So another question in the queue here, if we apply for and receive a stage 1 award, is the project eligible later for stage 2 and 3 as the project progresses?
Yes, absolutely. So again, this is be our first global round. So what that exact process looks like, we need to figure out a little bit, but for sure, we would love to shepherd something to IND, or if there's at an earlier point a handoff to a pharmaceutical company, then that's a win as well. So we would, if the project is making a progression, we will definitely allow it to move to the next stage. In fact, I can envision, again, this is me hypothesizing about the future, so don't take this as law. But if a project goes really well and really, it's a stage 1 project, and after 6-7 months, we're like, man, this is going like gangbusters, then we will try to figure out to kind of morph it into a stage 2 and get additional funds to you without having to wait till the next deadline. Because it's, for us, as well as your university, also an advantage to, get going as fast as possible because your patent clock is ticking. So and plus, of course, patients are waiting, right? Let's not forget about that either. So we will not make you arbitrarily wait for the next cycle. And most likely, this is just me, spitballing here. But for sure, we would love to support you all the way to the clinic. But ultimately, I think our ultimate KPI, key performance indicator, is to get something to patients. But a more proximal one is, will we get anybody interested that wants to take it and run with it? And that's really where that, shaping what that data package looks like, knowing what the regulatory strategy is, having that laid out, will really increase the chance of a licensing event. Or, startup formation with venture capital being interested in funding this.
Excellent. And I think we have time for one more question, and this is clarity on co-funding. So if funding is received for one aspect of the project, TRxA fund different aspects of the project, or are we looking for co-funding for the project as a whole?
I think both could actually work. That's the advantage of being new as well, that we get to make up the rules. And I think it really depends to who the other funding party is. So there may be a little bit more set in stone rules for, let's say, governmental funding from, let's say, the European Commission or FDA or DoD, something like that, versus a patient foundation who also wants this project to get to patients as quick as possible, whatever their funding model is. And we can be flexible. And that's the nice thing too about a Critical Path Institute. We're about 150 employees or so now. We're small, relatively speaking. We're nimble, we're flexible, so we can work out what is the best for the project and also with the other co-funders and what their requirements are. We'll probably need to work out some reporting, but that's all manageable administrative paperwork that we can easily figure out.
Great. Okay, so we have just a minute or so left. So Micah, I want to thank you for this great overview and for answering everyone's questions. Very, very informative. And I also want to thank our attendees. Thank you for your engagement. The questions were coming in fast and furious. I'm typing some answers in, so I will keep doing that if you haven't received an answer to your question. And again, if we didn't get to your question today, please reach out to us. The email address is right on the screen here. You can reach out to us with your question and we will get back to you. You can also reach out with a request to be added to our mailing list for future news and updates. And we do also, you can follow us on LinkedIn. We post there as well.
We have a video in the works. It's going to be a cool video. It's like 2.5 minutes. So you can, watch it in between meetings and get in 2.5 minutes in visuals what we try to do. So, yeah, Michelle, you had the harder job. Thank you for moderating those questions. All I had to do was, think, and you had to actually read through the questions. So yeah,
it was fun. Yeah.
On my behalf too, for everybody who attended and asked questions, thank you so much for being here. But we're here to help, so please reach out.
Thank you, everyone.
Great.
Bye-bye.
Bye-bye.